Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, Dovila Gruzdite, Global Product Manager for Cell Line Development from Cytiva.
Thank you, Alex, for that introduction. Good morning and good afternoon, everybody. Today, I would like to talk to you about ELEVECTA, which is Cytiva's cell line development platform for creating a truly stable and inducible producer cell lines for large-scale AAV manufacturing. Here in Cytiva, we truly believe that ELEVECTA is a unique producer cell line that is different to other assumed producer cell lines out there on the market. And today I would like to highlight that uniqueness to you. Gene therapy industry has a long history and it has evolved over many years with its different shortfalls and fallbacks, and then with some groundbreaking achievements. Today we are talking about AAV exclusively because in Cytiva we believe that this process of AAV manufacturing still has a lot further to go for a fully productive and very well optimized manufacturing process. AAV is still a dominating viral vector type for gene therapies, and this is where we have focused quite a lot in the recent years for different manufacturing techniques, optimizing them, as well as different equipment optimizations that we have developed. So we continue in that direction even today. What is really important to really start take notice of is that gene therapy is no longer a rare diseases only treatment method. Gene therapy has quite strongly entered the prevalent indications sector. And as you can see in the figure on the right, prevalent diseases have taken over rare diseases clinical trial numbers by quite a lot. And that means that different successes in the past have really built up the momentum and the confidence in the gene therapy community to start investigating different treatments for prevalent indications that will have a much bigger impact and bigger reach to these patient populations. What you can also see in this graph is that phase 3 has also got a very strong pipeline for prevalent indications versus the rare indications. And that means that we're really close to the point where therapy for these prevalent indications will be commercialized. And that means the demand for material for such treatments will be much higher than for rare diseases. And this is very important to realize and take notice of because science in gene therapy is really going full steam ahead. The manufacturing of these therapies is really lagging behind, and majority of gene therapy manufacturing methods are still relying on the monoclonal antibody production methods. So we still have a lot further to go to be able to really scale up our processes to produce this material that's required. And here in Cytiva, we know much better than anyone else what challenges our customers are facing when producing viral vectors. Starting from upstream and all the way to downstream. And we have focused for many years, and we thought long and hard on how to change our equipment, how to make it much more suitable for gene therapies. However, we have come to realize there is only so much you can do in downstream to save and purify this really precious product you created at upstream. And where the real potential lies is the biology section, where the process actually starts. So in Cytiva, we have started looking at the cell lines where the host cells are actually producing the AAV. And this is where we want to really maximize and really unlock the full potential of these cell lines, such that our downstream process can only help us to recover as much of that product as possible, not to actually help us meet the demand of the material that's required. Today I would really like to discuss different manufacturing methods of AAV from the cell line point of view, to be able to show you where we in Cytiva believe is the true future for large-scale manufacturing of AAV for prevalent indications for large patient populations and high systemic doses. So on this slide, you can see, let's say, 3 or 4 main methods of AAV production. On the left is our state-of-the-art triple transfection, whereby 3 plasmids carrying different genes have to come together inside the host cell to be able to produce your AAV of interest that carries your gene of interest. And this process has a lot of advantages, such as flexibility and cost, and the speed at which you can manufacture. However, it is highly unreliable, uncontrollable, and it doesn't provide ability to a robust, scalable, large-scale manufacturing process. And you can see going into the packaging cell lines, this as a kind of an evolution of this method, whereby people have tried to stably incorporate at least the 2 or 3 elements of that are required for AAV production. So you can have your adenovirus and rep and cap genes, adenovirus helper function, and rep and cap genes incorporated stably in the cell line, and all you have to add is the gene of interest. Or you can have your rep and cap stably integrated, and then you have to add your gene of interest and your helper virus. But this carries different disadvantages for viral cross-contamination, and also you still have to produce your plasmid of interest. So going then towards the alternative manufacturing platforms, such as baculovirus with SF9 insect cells, people were able to dramatically increase the yields, and we even had a successfully commercialized serum on the market, Glidera, based on baculovirus. However, these cells pose immunogenicity risks to the patients, because there is a chance that different proteins from insect cells are incorporated into the capsid of the AAV, which then are not recognized by human body after the therapy is administered. So the way that is really most preferred and where we want to go in the future, and this is simply a matter of time, we believe, is where the industry will shift to the true producer cell lines that are delivery free, and they carry 0 cargo. So you have your helper virus, your rep and cap genes, as well as your gene of interest stably integrated in your host cell, and you are ready to manufacture every single time, every single day before your batch, with a simple step of induction. And that is exactly what ELEVECTA is. So here in Cytiva, we were able to develop a platform which allows us to help our customers to take their therapeutic concept and bring it into the producer cell line and then manufacture the therapy. So in Cytiva, we start the process with ELEVECTA alpha cell line, which is our proprietary cell line where rep and cap genes are stably integrated. We then ask our customers to provide their cap gene as well as the transgene. And as a first step, we create a polyclonal producer pool, out of which we then start with a single cell cloning process where we screen a different panel of clones and we isolate the best performing clone. And this is the producer cell line that we hand over to our customers. Upon receipt of these cells, the customers are able to create their GMP cell bank and manufacture. So as I mentioned earlier, you can start with your small flask, and then you can seed a smaller bioreactor and then take it to a larger bioreactor, induce the cells there, and you start your AAV manufacturing. As simple as that. There is zero transvection required at manufacturing stages or no helper virus is needed, just your producer cell line. What is also very important to recognize here is that the optimization process is key. So going from the producer pool to the single cell clone to the fully optimized upstream process, we're able to elevate our viral genomes per liter by nearly 2 orders of magnitude. And that is where Cytiva can really help customers. We have done some scale-up studies on our ELEVECTA platform in the 200-liter Paul Allegro SDR bioreactor. And from the data, you can see that the cell concentration and cell viability is improving, going from 10-liter bioreactor to 200-liter bioreactor. And then on the figure on the right, you see that the titers going from 10 liters to 200 liters are really comparable, which shows the robustness of the scale-up of the ELEVECTA producer cells. So thank you everybody for watching this presentation, and I'll be happy to take any questions.
All right, great. And as a reminder to our audience, you can go ahead and type in your questions for our speaker now. So our first question is, does ELEVECTA work with any AAV serotype and GOI?
Indeed. So we take any serotype and any gene of interest within the limitations of the AAV, and we work with a customer to optimize the process for the gene of interest and for the serotype. And then going through the development process, we make sure that we pick up the most important characteristics from the customer for the single cell clone as we do the screening.
And our next question is, in your presentation, you mentioned increased titers for optimized process. What does that entail?
Excellent question. And this is where we want to really emphasize the importance of a holistic view of this upstream process. So the cell line isn't there to take off all the brunt of high yields and excellent performance. There is an element of lots of different things coming together. And this is where in Cytiva, we believe we can really help our customers. We have media development services. We have excellent application specialists. I can go to the customer site upon the tech transfer of the cells and really help you work with the cells at a large scale. And then there's also the process development at large scale that needs to be optimized to really get the most out of the cells. And having this in a holistic and all-around way, we can really then maximize each customer's gene of interest production. And this is where we believe the future of manufacturing lays, is that we do all the optimization at the beginning, and then all you have to do with every batch is just produce.
Okay, great. Well, it looks like we're running a bit short on time, so if we don't get to your questions, we'll be passing them along to Davila, who can follow up with you directly. So please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, how are HEK and CAP ELEVECTA platform different, and why would some customers choose one over the other?
That's a good question. So ELEVECTA platform is an acquisition of Cytivus from a company, Sevec. And CAP cell line is the proprietary cell line that the company was really built on. So as a legacy, we have two cell lines to choose from. The customers have a choice to go with two cell lines based on their performance, because we know that different serotypes, different genes of interest will express differently in the two cells. So we are proud to be able to offer ELEVECTA platform on two different cell lines, and then the customers can make up their mind based on the performance of the two.
Okay, well, thank you, Davila, and thank you to our audience for joining us. The recorded version of this webcast will be made available for on-demand viewing on our website, and as a registered attendee, you will receive a follow-up email providing you with a direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.